that he was probably wasn't gonna be able to make it, so he wanted it recorded.
Good. Well, we automatically record, but I like to make sure that, you know, there's nothing here that you'd be nervous about us recording.
Oh no, I'll just be baseline nervous giving a talk, don't worry. Other than that, shouldn't be any weirder.
Happy Friday, everyone. 4 more weeks left of classes.
Just that many.
Baby.
Wow. I didn't realize the semester was so close to being over.
It's really wild. I mean, so the semester started so long ago, it feels like, but the students weren't physically back, or most of the students weren't physically back until Labor Day. So it's sort of, you don't realize how much of the semester happened before they got here. So
yeah, that's crazy.
Yeah, pretty wild.
We'll see how the school handles bringing them all back after Christmas then. All right.
Yeah, I mean, I think we're starting normal time. So we're starting kind of like January 20 something. For a semester, obviously, us researchers just continue our normal lives. But so it's almost like a summer. I mean, it's like a 2-month break because they're gone by Thanksgiving.
They're not going to bring them back after Thanksgiving, are they?
No, classes end the 13th, and then we have finals, and then they go home for Thanksgiving and don't come back until January 20 something.
Wow.
So it really is like a summer break. It's long.
Yeah, it's a lot of time.
Yes, it is. I'm so happy. It's exactly 12 o'clock, 12:01. We'll give it another 2-3 minutes just because, you know, connection issues and whatnot.
Sure.
Look at that little doggy, Jen.
Really excited for Biomath.
Yeah, totally ready.
This is it.
Yeah.
My cat is having like a field day right now. She's like bouncing off the walls.
She's excited for your talk too.
She's just like, why can't we play?
Does this look like an excited face to you though? He looks pretty sleepy. How are your cats, Helen?
Well, they live at my mom's house, thank God. They're fantastic. They are so sweet. They ended up being like really affectionate, snuggly cats, which is sort of perfect for the kids and the parents. They just all do their thing. Just very lovely. All right, well, it's 12:02, so I guess we can get started. I will announce today that we do have a talk in biology. I don't know how exciting it would be for biomath, but for the people into ecological and evolutionary theory, there's going to be some of that. It's a talk about the breadth of hosts for different viruses. So this is a bacteriophage system, but it's some pretty cool work. What else? Any other announcements? Nothing? All right, then we will hand it over to Daniel today to talk to us about some of his dissertation work.
Yeah. So today, thanks for having me, everybody, at the Biomath, and thanks for coming to my talk. Yeah, today I'm going to be talking about the research that I've been doing last several months, almost a year now, with this group. I can go ahead and start the slideshow, I guess. With this group, it's me, Christopher Del Negro, Gregory Conradi Smith, and Cam Grover does a lot of our wet work as well, wet lab work. Where we're interested in creating a model for eupnea rhythm and also specifically the Psi rhythm to understand how they work and see if we can't learn something from it. So we'll get into kind of what those words mean and whatnot. Yeah, let's begin. So this is going to be kind of the overview for the talk, the roadmap that we'll refer back to. Shouldn't mean much to you yet, but hopefully by the end of the talk, you'll understand what each of these bullet points are. The general idea is that first, I'm going to show our creation of the eupnea model, mathematical model, and then I'll show the Psi model, and then we'll bring them together. And if there's time, we'll show some of the applications we've done with it. So yeah, before we get into all this fun stuff, we'll have to do a little bit of background on what our lab actually does and what those words are. So in the Del Negro Respiratory Lab, we're interested in breathing, specifically where in the brain that rhythm for breathing comes from, and even more specifically, how it's generated in the brain. So I'm showing here a plethysmography setup. So this is a plethysmography chamber where we would put a live mouse in, and the mouse just sits in the chamber and breathes, and we pass a known volume of air past the mouse, and we can record changes in the pressure from a reference chamber and the recording chamber. And those signals look like what's here on the right. So we can see volume flow changes here, which correspond to just normal breathing for the mouse. So if I say something like eupnea, or if I say inspiratory rhythm, I'm talking about just normal breathing, just unlabored breathing that's going on. I call it inspiratory breathing because, in fact, the muscles are only really being recruited during the inspiratory phase of the breath. So that's this, this little rhythm here, the mouse breathing. And as a lab, we're very interested in where this signal comes from and how it's generated, exactly what the mechanisms are. But for this project in particular, our group is interested in these larger breaths right here, which are known as Psi breaths. A Psi is about 2 to 5 times the size of a normal breath. It's just kind of what you think of as a deep breath. And it helps to reinflate the lungs and to essentially, there's a little bit more detail there about exactly what it's doing, but just know that they do serve a pulmonary function to keep everything working. Like I said, they're much larger. The other ways to identify them is that they're very regular. So they are a lot slower than normal breaths. They're about an order of magnitude slower, but they are very regular, in fact. So on the order of, say, every few minutes. Other characteristics about a Psi breath is that they're often coupled to the normal breathing rhythm. So when I say that, I mean that if you're having a normal breath, a Psi tends to happen right on or sort of piggyback off of a normal breath. So if you look at a lot of these, the Psi seems to sort of know when to appear. It kind of pops up right where a normal breath would be. So that's sort of like the eupnea rhythm, somehow communicating to the Psi rhythm. And the other way, after a Psi, you'll notice that there's sort of a depression in the breaths. There's like what we call sometimes a post-Psi apnea, where the normal eupnea rhythm actually slows down. So somehow these rhythms are communicating with each other. I'm just going to show here, this is another plethysmography recording, but now it's very zoomed out. So now the eupnea is just these little peaks here, and the Psis are these really tall peaks. And this just kind of shows you that they are very regular on the order of a few minutes. In this case, like 2 minutes for a mouse. Okay, so that's the behavior we're interested in, eupnea and Psis. And now let's dive deep into the brain to see where they're coming from. So this is the parasagittal view of a mouse brainstem. Sideways like this, if you can see my video. And I've highlighted, some anatomical features have been marked, but most importantly is the one in yellow, which is the pre-Botzinger complex. This is going to be the area of the brain where the inspiratory rhythm it comes from. And not only that, not only does it generate the inspiratory rhythm, it's also going to be generating the Psi rhythm. And I'm going to show that in just a second here. But just to continue, so if I showed, in the last slide, I showed you the in vivo approach. This is something our lab does to study breathing in the normal in vivo approach. The mouse is alive and breathing. I'm going to show our in vitro slice preparation. So this is the other way that we're going to kind of study this rhythm in the brain. So what happens is, as I said, this is the parasagittal view of the brainstem. Right where that gray bar is, we'll come in and we'll just take a slice about 0.5 millimeter thick of that brain, and we'll kind of imagine pulling it out of the screen and turning it and laying it flat. You get what kind of looks like over here. So you can see a lot of those anatomical regions are still captured, including the pre-Botzinger complex. This is another view of that slice preparation, again, about 0.5 millimeter thick, and red now is the pre-Botzinger complex. And what you can do is you can take an electrode and you can just drop it right on top of the pre-Botzinger complex and record. And you can get electrical activity. So that tells, and you can actually, I didn't show it here, but you can actually just cut away just this red box. It's called an island preparation. And you'll actually capture this rhythm here that is known to be the origin for the, in this case, these little bumps are the origin for the inspiratory rhythm. And these much larger bumps are actually the origin for the Psi rhythm. So it's pretty fascinating that this group of neurons, if you cut it away and just recorded from this little area here, somehow the rhythm is still being generated. The cat is like freaking out. The rhythm is still being generated. And what we're interested in is how this little cluster of neurons is able to generate a fast, low amplitude rhythm, that would be the inspiratory breaths, and how it's able to generate a Psi rhythm as well. Another thing to point out before I move away from this figure is that, you know, the size is maintained from the size of the Psi is maintained in this in vitro preparation. The regularity of them is maintained as well, and also the coupling. So what we typically see is these, I'd say these are the three kind of most common traces where the Psi will sort of appear right where you'd expect a eupnea to appear, and then you get this, what we call post-Psi apnea, so a lapse of activity for a little while. This slide here, I just want to kind of show the similarities between what we find with the plethysmography in vivo, this sort of recording, and then what we actually see when we do electrophysiology in the slice preparation. Very similar. We can see eupnea rhythm kind of translates and the Psi rhythm as well. Okay. So now I just want to kind of point out, as I said, you know, we're really interested in how these rhythms are generated. And I think that the three most important questions of my PhD, I think, are going to be, how is this inspiratory rhythm generated? What are the details behind it? How do the neurons do it? How is the Psi rhythm generated is another question. And importantly, what mechanisms couple them, which I think we'll have to learn as we sort of understand how these rhythms work. And in particular, this project is going to be focused on really question 2, how's the Psi rhythm generated? And as a byproduct, I think, of that question, we'll have to answer things like, or hypothesize about what couples them as well. So a little bit question 3. So before we get into the model and stuff, well, what is known about the inspiratory rhythm and the Psi rhythm? So it's been pretty well established in the field at this point that the inspiratory rhythm is the result of a group of glutamatergic interneurons that are wired up in this network where they sort of excite each other. They're all excitatory, and so there's sort of a buildup of excitation in the network that brings about like the upstroke of the burst. And then there's some sort of activity-dependent mechanism, so it could be something like synaptic depression or the recruitment of an activity-dependent outward current that terminates the burst and shuts it off and restarts the whole cycle and initiates this, the refractory time between events or something like that. So the whole process repeats again. On the Psi side, a lot less is known about the details of the Psi rhythm, but there has been some evidence to suggest that it is related somehow to intracellular calcium dynamics. So if you play around with the calcium handling inside the neurons, inside the cells, you can disrupt the Psi rhythm. So there's some evidence to think that it's somehow calcium related. And what we're going to present in this model is sort of a full mechanism for how the calcium handling might actually be being carried out to create the rhythm. And I leave these arrows in here as well, just to remind you that there is some sort of coupling between these two oscillators, and hopefully we can start to hypothesize about what those arrows are. Okay, so that's it for background. So now we're going to move into the first part of what we've been doing, which is building the inspiratory rhythm, building a model for the inspiratory rhythm. So we adapted a model from this paper, I believe it's a book chapter actually, from Tabak and Rinzel in 2005. And I'm just going to need to do a quick kind of explanation of what exactly these variables are. So there's only going to end up in this first simplified model 2 equations, two variables. One is the average network activity. So sometimes this is called a mean field model. It's not a model of a specific neuron. A is just going to reflect the average activity of the network. So its units are actually spikes per neuron per second. So it's going to be the average firing rate. And then this S term is going to be the average synaptic depression of the entire network as well. Okay, so those are the state variables of the ODEs, the 2 ODEs. And then I just want to kind of give a quick preview of what the model's eventually going to do before I get into the actual mechanisms of what's going on. You can see activity levels sort of building up, and then once synaptic depression relaxes enough, you have a spike. Synaptic depression immediately kind of shunts the activity, and the activity drops back down, and synaptic depression starts to recover since there's no activity in the network. And this process just keeps going on. Okay, so let's get into a little bit of the details about exactly how the model is generating this rhythm. So the first thing I want to kind of point out about these 2 ODEs is they're really not too crazy. They're simply saying that these variables, A and S, will relax to some, what ends up being a sigmoidal line here defined by either A infinity for A or S infinity for S. You can see that these variables are going to relax to these sigmoids, sigmoidal curves or lines, at some characteristic time constant. So they'll relax to some curve at some time constant. That's all that these equations are really saying right now. The other thing to kind of point out is this little funny term here, which is really just the noise for the system. So it ends up just being Gaussian white noise into the model. It just injects a little bit of stochasticity into, say, the activity of the network. So just random firings, or just sort of a little bit of randomness into the network. So that's what's going on here. And now I want to just kind of actually plot what this A infinity curve looks like as a function of A. Let's take a sip of water. Okay, so as I said, A infinity is really going to be defining sort of what you would where you would expect the activity A of the network to sort of relax to. So A infinity is sort of describing what the network would want to be firing at, the firing rate of the network, given a W in this case is gain, so just like the average strength of the connections between the neurons, sort of a knob to turn up or down, just average excitability. S, which is the synaptic depression, right? And then whatever the firing rate of the network is at that moment, all these things, you can call them sort of like the average synaptic drive of the network, will determine what the rest of the steady state activity levels should be. So that's really what this is saying. And we can look at it a little bit more closely. So, we set, say, W to 1, I think in this case, and we ask, what does the network want to do? What does the A infinity curve look like for different values of A and S? So as a function of A and S, the simplest case, I think, to look at first would be to ask, what does the network do when, say, synaptic depression is off? So when S is equal to 1. You can see when S is equal to 1 in the equation here, it really just goes away. You end up with just W and A. So synaptic depression being completely off, just not really, there is no synaptic depression, is like S being 1. And when that happens, it's this dotted line here. You can see that the network for any activity level will try and relax to some higher state. Eventually will relax to some, you could say, the up state or the maximum state of the network. That's where all the neurons will be firing at their max firing rate across the board, which sort of makes sense, right? If there's no shunt on the network, if the synaptic depression is off, then a group of excitatory neurons with enough, with enough gain should eventually sort of through positive feedback loop enter some sort of high activity state. Okay, so conversely, what happens when synaptic depression, when the synaptic depression in the system is very high and the system is very depressed? So in this case, we could set S equal to 0.2, and that'll give you this dashed line down here. And now you can see that for any value of A, for any firing rate of the network, the network will tend towards a state of lower activity. And so this will eventually pull you all the way back to a down state. So if the synaptic depression in the system is very high, then the network will eventually stop. And now you can see that there's these two possible configurations for the network to be in. And if we allow the activity of the network, A, to change very fast, and we say that the synaptic depression, S, is going to change very slowly, then we can enter a system where the activity is going to be jumping up, and then synaptic depression needs to slowly catch it up to it, turn it off, and then the whole system will repeat like that. Another way to kind of look at it is to go ahead and a lot of people do this. They'll take their ODEs and they'll try and find the null lines, set the derivatives equal to 0, and solve for the steady states of these values, and you end up with their null lines, which like I'm showing on this graph here. So the null line for A ends up being this little cubic curve here, and the null line for the S term is this light green. And then you can see in this setup that we're going to end up with what ends up being a limit cycle. So A changes very rapidly. It comes down here, and it's fixed to the A null line, and the synaptic depression term, S, slowly recovers. It's being pulled towards the null line for synaptic depression. And as it continues, the synaptic depression continues to recover. Eventually, you're going to fall off this knee here. And now the steady state for the activity is up. And so this whole trajectory kind of jumps up. And as the activity builds up, synaptic depression now being pulled back towards lower values means that synaptic depression is kind of gaining up in the system, and the trajectory eventually passes this knee up here, where now the new steady state is back down at the beginning again. And so this whole system kind of repeats itself. To kind of draw it up to what's happening up here, this down state here, it corresponds to kind of the intervent interval here. And then once you fall off this knee, that's this upstroke of the trajectory, corresponds to upstroke here, and then it falling down here, and the whole system repeats as well. So you can kind of see where those two things are connected. One additional thing to note here is that the timing between the events is very regular, right? Just something to note. Despite the stochasticity, the distribution of the events appears to be pretty normally distributed. And then in detail here, something to kind of look, if you look at closely, and this is what they talk about a lot in this paper, is that there's something interesting going on here with the way that this model is working with these two variables right now. And that's the fact that if you follow this trajectory here, when it falls off this knee, it sometimes comes off a little early and sometimes it comes off a little late. And as a result, the synaptic depression has either recovered more or less. And what's going to happen is when the trajectory comes off this knee, if it comes off early, the entire rotation around this little limit cycle ends up being smaller. And if synaptic depression recovers more, the trajectory is larger. So what that says is when synaptic depression, it doesn't recover as much, the next burst is going to be smaller. So there's a correlation between the duration of the inter-event interval and the size of the next burst. So it makes sense, right? Synaptic depression is allowed to recover, the burst is larger. Another way to kind of show that is doing this little modeling study here. What Greg did was he actually perturbed the system while it was recovering. So while synaptic depression was still very much recovering, he perturbed the system over the threshold it needs for a spike, but the spike is very small, right? Synaptic depression immediately just kind of can't really do much. The system doesn't really get very large of a burst. What the problem with what we're seeing here is that this isn't exactly what we see in the Plethysmography complex in vitro. So I'm going to point here to a study by Andrew Kotick and our very own Christopher Del Negro in 2015, where they're going to use optogenetics to excite the core rhythmogenic neurons of the Plethysmography complex. So they know they've got a good marker for the neurons that are part of the rhythmogenic core for the inspiratory rhythm. And what they can do is they can actually goose the system up enough to get a burst. And what they're showing here is that you can't necessarily get a burst right away. There's some sort of refractory period where the system goes through, and after some amount of time, now when you kick the system, you get a full burst. But what's really important for us here is the fact that when they do get a burst after waiting for some refractory period, the size of that burst is always the same. The amplitude and area are always the same. And so there's no graded response, which tells us that whatever is sort of recovering during this period has completely recovered by the time that there's the next burst. In the case of our model, that would be like, okay, synaptic depression has to recover all the way. The synaptic depression should recover all the way. That way the next burst can be a full size. So we can put the model into that configuration. You just play around with the parameters a little bit until you get a phase diagram that looks like this. Now the knee that normally the trajectory would kind of slip off of that knee, now it's being, that point is sort of held in front of a steady state for the system. So the system will just stop and it won't fire by itself. Now what you're waiting for is this noise term to push you over some thresholds that you can get a burst or this trajectory to go around. The issue here, right, is that now you're waiting for a random event. You're waiting for noise to push you over some threshold. So it's very similar to a Poisson process, or waiting for a random event that has some average chance of happening. And as we know, when you're waiting for a random event to happen, the timing between any given event is going to be exponentially distributed. And I think we all know that our breaths are not exponentially distributed. That would be mildly terrifying if that was the timing between our breaths. So the model in this configuration doesn't quite work either. So this led us to the conclusion that we likely need this third variable, something else going on in the system. And this was also presented a little bit in the Tabak-Renzel paper. So we're, again, just taking another page out of their book, where now you introduce this, what we'd call maybe cellular adaptation, which acts as like a threshold on the synaptic drive. So there's going to be this data term, the cellular adaptation, is some threshold that's slowly dropping over time. So it's like a time-dependent threshold that gets easier and easier to overcome as time goes on, right? And so what it ends up looking like in the trajectory here, I think this is the easiest way to see it, is that we have our activity up here bursting away. Synaptic depression is what's terminating the bursts, and it's allowed to fully recover. By the time the next burst comes, the synaptic depression is almost always recovered. And what's really triggering the burst now is this other sort of threshold term, or cellular adaptation. And I won't go into what ends up being a null space now for the A infinity curve. I don't think we need to do that. I think it's just important to see that it's theta now that's that is triggering the burst, and it has no effect on the size of the burst. And the events are normally distributed. So that is, I thought I had one more thing to say about this. Oh, I just wanted to say, if you're a little confused by what cellular adaptation is as a mechanism, I think that's okay. I think at this point, right, the whole point of this model was to study what's happening or how the psi rhythm is generated. And so it is sort of a bit of a question mark, which I think we're happy with, right? I think after we're done with this project, the next kind of project is going to be, well, what is cellular? What does this term actually mean for the cell and for the network? So we have some theories, but we won't go into them now. So that is basically part one, which was how we generated the model for the inspiratory rhythm. The next part is going to be how we generated the psi rhythm. So before I go on, any questions on what I just showed? All right, cool. Yes, so the next part, how to create a model to generate calcium fluctuations that could generate long, large period oscillations. So before I get into the details about the equations and stuff, there's some nomenclature, some things I have to kind of get out of the way. So we're talking about calcium concentration, intracellular calcium concentration in the cytosol. We're going to talk about calcium concentrations in, I'll probably say ER, but it also means any sort of calcium sequestering organelle that could be, have a calcium concentration that's taking calcium away from the cytosol. And then that means that there's some total calcium in the cell. That's the combination of the cytosolic concentration and the ER concentration. Now, the way it's going to work out, I'm just going to be talking about the total calcium concentration and the cytosolic calcium concentration. It's going to end up being easier in the long run when we start doing the phase plane analysis and stuff, if we only keep track of total and cytosolic. But at any point, because you already know two of the values, you can always get the ER concentration. So I won't exactly be showing this variable, but it can be deduced from simple math. Okie dokie, let's do this next slide. Okay, so I'm gonna give a quick preview again of sort of what the model is gonna end up looking like, what we want it to look like, where everything's running here. We can see this will be cytosolic calcium changing with total calcium, and now we've got these really far away, spaced out events. Okay, so how exactly do we do that? So we borrowed a model from this paper, Kaiser and Levine, 1996. Okay. And it looks a little crazy, but I'm going to break it down piece by piece. Yeah, 1226, perfect. I'm going to break it down piece by piece, and I think it'll make sense by the end about what each of these elements actually are. They're not too complicated, and so I think you'll see what each of them are. Starting with what I think the simplest one is oh, by the way, before I begin, obviously this is the cytosolic concentration, changing cytosolic concentration. So all these terms are going to be talking about changes in the cytosolic concentration, and then total calcium concentration. So not the internal changes, just what's happening over the cell membrane. So the first term I want to explain is this J0 term, which is just passive calcium uptake into the cell. So any calcium that makes its way into the cell just passively is described by this J0 term. It'll probably be sequestered right away into the ER, but that's not really being, that's not the point yet. So J0, just calcium intake. This J1 term, which you can see is dependent on activity. This is calcium that comes into the neuron anytime there's some sort of activity, right? If you think about calcium coming in during a spike or something like that, that's calcium uptake as a function of activity or dependent on activity. And then we have these calcium extruders across the membranes. Just the cell likes to keep the calcium concentration low in the cytosol, and so you have calcium being pushed out of the cell. Okay, and the next one I'm going to point to here is in blue, this V1 F infinity as a function of C. This is talking about when calcium binds to IP3 receptors or ryanodine receptors, and it initiates calcium release from the ER. So if the calcium concentration is just right, you'll get calcium being dumped out of the ER. And I'm not going to show the FFA function. It's basically a bell curve. So at some low calcium concentration, there's not a lot of calcium efflux. At just the sweet spot where the calcium's just right in the cytosol, you can get calcium dump out of the ER. That's this V1 term here. The next one is V2, which is just calcium leak out of the ER. And both of these things are multiplied by some driving force. So this is, the difference between the cytosolic concentration of calcium and the ER concentration of calcium. The larger that difference is, the larger this drive, the larger both these fluxes are going to be. And the last one in purple here are circa pumps. So the pumps that grab calcium out of the cytosol and put it right into the ER. And I know at least one of you is thinking, well, what the heck with all these channels? Why aren't these channels and pumps? The drawings, I did these drawings, I didn't have time to change it. So they're not all channels, right? Some of these are actually pumps. So if the images are bothering you, I understand, and I wish I'd had time to change it, but I did not. So cool. Okay, so what exactly is going to happen in the model? Well, why don't we set the derivatives equal to 0 and find the null lines? And now you can see why I am looking at total calcium here. It ends up, the null line ends up being just being a straight line, which ends up being completely dependent on the J naught term, which is really nice. You can move this around and see what happens. And I don't think I mentioned this, that the calcium fluxes intracellularly, so across the ER to the cytosol, these are all, we're just going to presume they're happening very fast relative to calcium fluxes over the cytosol, over the membrane for the cell. And if we make that assumption, then we can get to see a fast flow subsystem kind of coming out here, where you'll have this limit cycle up here, which I'm going to show in context with the actual time series. I think it'll be easier to see it this way. So calcium concentration in the cytosol is very low here, but just through calcium kind of leaking into the cell and immediately being uptaked into the ER through circuit pumps, there's a buildup of calcium going on in the ER, right? If total calcium is changing, but the cytosol calcium isn't really changing that much, then it must be going to the ER. So that's this here. And then at a certain point, the calcium concentration in the cytosol and ER get to the right kind of ratio, the right concentration, where now calcium is going to get dumped out of the ER into the cytosol, and it's, that'll give you this huge jump up here of calcium in the cytosol, and then it's immediately kind of pumped out of the cell by the calcium pumps, moving it out of the cell. And so you reset back down to a low concentration in both the ER and the cytosol, and then the ER begins to kind of take more calcium up again. Okay, so this is the oscillation for that kind of describes the calcium dynamics that are happening in the cell. Calcium buildup in the calcium buildup in the ER. When it gets to a certain point, calcium initiates more calcium release from the ER, and you get this huge bump up into the cytosol. So that's all there is to it, to the calcium model. And now, like I said, we want to bring these two models together. So other than just writing all 5 equations down, I think what's important to note here is how we're connecting them. This gets back to how are the two oscillators coupled. So first, I'll point out, how does the inspiratory rhythm, the eupneas here, how do they talk to the Psi model? Well, that's through this J1A term here. Remember, that's calcium coming into the cytosol as a function of the activity. So if there's spikes, the cell's really active, then there's going to be calcium influx into the cell, and that's this term here. So that's how the eupnea or inspiratory rhythm is talking to the Psis. And then back, how does the calcium fluxes, how do they change things like the activity level? And that's going to come in through this A infinity function. So whatever like I said, what the steady state for the activity should be is now not just dependent on the synaptic depression and things like the cellular adaptation, it also depends on calcium. So if calcium concentration in the cytosol is very high, then the steady state for A can be much higher as well. And so it's going to want to move towards that higher value, and that'll give you this big burst here. And then it ends up being this overexcitation of the network that drives things like synaptic depression way down. It ends up being very depressed, but the number goes very low. And then also for the theta, like the threshold, it gets pushed up very high, and now you have to wait a little bit longer for it to get back down to this level where you can have activity again, and that'll give you the post-Psi apnea. So that's the model. Yeah, we like it a lot, and we worked on it, and I think it does a pretty good job. And now what we want to do is see what we can do with it. So I actually have plenty of time. So I think I'll get into one, maybe 2 of the applications for the model. But the first one that I'm going to talk about is, before I go on, any questions on the math that I just talked about before I get into how we are going to apply it? Okay. So one experiment that somebody did in this Lee Nature paper 2016, where they're going to actually be modulating the Psi frequency. So this here is in vivo. They're injecting this drug called, it's not a drug, it's a neuropeptide called neuromedin B directly into the pre-Botzinger complex. And it binds to neuromedin B receptors that have been shown to be in the pre-Botzinger complex. And they find that this is in vivo now, the mouse Psi rhythm actually speeds up. So differentially increases the Psi frequency. You can just see it here, and this is like the plethysmography trace again. Here they're actually recording from the diaphragm, so you can see the muscle innervation. And here they're just showing the frequency peaks 17-fold. So that's in vivo, and I just want to do one more thing from the paper. This is, they actually also do it in vitro now. This is like the slice preparation where they're gonna just dump the NMB, neuramine B, right onto the slice, and they show in slices as well when you add this bombesin-like neuropeptide, neuramine B, you can increase the Psi frequency. So we looked into it a little bit. And it's known that this neuramine B receptor is linked to the GPCR, G protein coupled receptor, specifically the Q-type GPCR pathway, which, just for a refresher, is going to end up activating phospholipase C, which will increase IP3 and DAG production, which will eventually, IP3 binds to IP3 receptors, initiating calcium release from the ER. So this can fit into our model really nicely because we have this term, like I said, V1 here, which is like, the calcium efflux out of the ER as a function of calcium binding to IP3 receptors or ryanodine receptors. And so what we did was we just said, okay, why don't we basically just increase this entire term, increase V1, what happens to our model? And so here's a control for some control V1. Again, I'm just looking at the subsystem now of calcium. I think I took it out of the entire other system for now. Just looking at those two equations, and we see that the rhythm speeds up, essentially. So matches really nicely with that finding of theirs. Something's in the chat. Should I see it? Let me check. Yeah. Okay. So it speeds up. Okay. And then if we wanted to look a little closely about why that's happening, the easiest thing to do would be to look at the phase diagram. So in red, I have the control NULC line for C, the calcium concentration in the cytosol. And what happens when we play with V, if we kind of tune V1 as if it was a knob, right? We're just turning it back and forth. What happens when we increase it? What happens to the C NULC line is it kind of gets smaller like this. And as a result now, I wish I had the trajectories here. Maybe I can draw them. Okay, so remember it was looking like something like this, right, before? And it spends the most amount of time in this downstate here, right? And then it kind of falls off this knee, and big influx of calcium into the cytosol, which is quickly dumped out. Remember, that's these little blips here are calcium efflux here. So it spends this whole downstate here is this little spot here on the NULC line. And so when you change V1 and you end up with a NULC line for C that looks like this, now the trajectory spends a lot less time on this downstate here. And it's even more obvious if I turn V1 the other way, where now I have a whole bunch of lines, but now you can see that it spends a lot of time on this downstate here. So it's nice to see the model working like that and recreating that result. Get rid of the annotations. What do we have next? So we have a couple things in the chamber still that I wasn't sure how much more stuff I was going to be able to talk about. But I think I'm going to just try and talk about one thing that we're doing right now in lab, which specifically, oh no, look at all that. Let's see if I can erase it. So I'm going to actually back up and show one of these plots that I showed very early on, which was just talking about these things as they're kind of their own oscillators, right? And I said that, it's these interneurons, excitatory interneurons that are initiating the inspiratory rhythm, and that it's probably calcium oscillations inside those neurons that are responsible for the slow Psi oscillation. But it's also possible, what if somebody asks, well, what if the calcium oscillations are actually happening and not in the neurons, but in the glia? And there's some other interaction between, the glia and the neurons that's responsible for the coupling. And it's a good question. And so one thing we can do is we can actually disrupt glia functioning. So excuse me if this is a little rough, but I didn't prepare too much to know if I'd have time for it, but I think it's kind of interesting to show. This is experiments that Cam's doing right now, where we added something called fluoroacetate. Fluoroacetate is going to get inside the glia, and it screws around with the glia-Krebs cycle and disrupts its ability to essentially make energy, and the glia will eventually kind of stop working. So it's effectively pharmacologically stopping glia from working. And then we're going to try and restore one of the things that we think the glia are doing, which is the glia are uptaking. They're uptake. Let me see if I remember this correctly. So they're they're uptaking the spent glutamate, repackaging it, and kind of giving it back to the neurons in order for them to, in the form of glutamine, in order for them to then turn glutamine back into glutamate and have more excitatory neurotransmitter. And so without the glia, there's not enough recycling going on of the glutamate and glutamate being turned into glutamine and then back to the neurons to be turned into glutamate. There's not enough recycling going on. The neurons kind of run out of things to pack the vesicles with. And so it'll eventually kind of turn off the rhythm, right? The neurons won't be able to communicate. And so what we'll do is we'll just dump glutamine right onto the dish, the slice rather. And this way the neurons will be able to take the glutamine in and convert it back to glutamate. So restoring what we think is sort of the role that glia has on at least interneuron communication. But if the glia are responsible for the calcium oscillations, then without them, without them being around, there can't be any calcium oscillations. And so maybe the Psi rhythm will go away. And so preliminarily, this is very preliminary, it could change. We still haven't really totally nailed down these experiments yet. But we can see that when we apply fluoroacetate, disrupt glia functioning, we see that this is the eupnea frequency down here on the bottom trace and Psi on the top. We see that there's a small change in the eupnea frequency. It tends to sometimes go down. I guess not really in these cases, but definitely the amplitude, which is shown by the color. So yellow being really, really high amplitude and dark blue, I wish these had the scale bar, really dark blue being very low amplitude. We can see it tends to lower the amplitude of the eupnea. So here going from yellow to dark, yellow to dark. And then on the Psi side, we're just going to be showing the frequency here. You can see that the Psis appear to stop. Now, whether or not they're just changing their shape and making it difficult to identify, or whether they're actually stopping, is still kind of the question that we're asking here. But preliminarily, it is very interesting that when we add glutamine in, we're not able to recover the Psi frequency, or so it seems. So a lot of questions still there to be asked, but this is just kind of hot off the presses, and I wanted to show some of Cam's work as well, since he's on this project more on the biology side than on the modeling side. Okay. Yeah, I think I'll stop there. I don't really have too much more. Give a thanks to everybody in the Del Negro lab, everybody working there who we bounce ideas off of, and then in Greg Conradi-Smith's lab, who I talk to those guys all the time about, different ways to plot things and whatnot. I mean, we talk about a whole bunch of mathematical stuff, so that's really good. And then the Biomath group, who I've been actually attending Biomath now since I was like a sophomore in undergrad. So it's been it's like 6 years or something. So I really appreciate this group and being able to present in front of it. It's a great exercise in presenting our work. So thanks a lot. And I'd love to field your questions.
Thank you, Daniel. That was fantastic. You did a really nice job of explaining the model and how the two are coupled. I will open it up for any questions. So,
Dan, I have a question.
Sure.
I may have missed this. Actually, I have a couple. Is the
Okay.
fluoroacetate specific to glia? And how does that work? And then the second one is, and I may have missed this, how does the Psi rhythm change during sleep and say during moderate exercise? Is there some type of pattern to this, or is that known?
Okay, yeah, so all right, I'll start with question one. Is the fluoroacetate specific to glia? As far as I understand, yes, it has something that is uptaked by the glia and disrupts the glia Krebs cycle and not the neuron or any other cells. I don't really know how it works, how it's specific exactly like that. I know that once it's in there, it's doing something where it's interrupting the process by basically getting in the way of one of the enzymes that thinks it has another protein, and now it can't break down the protein that it thought it had, and so it gets stuck. And I think pretty sure that's the way that works. But exactly how it's specific for glia, I actually don't have a good answer for that, but I'm pretty sure it is. Yeah. And then so the second question was, how does the Psi rhythm change during things like sleep and exercise? I wish I had a better answer for that as well, but I don't off the top of my head. I see something from Christopher. Let's see. Oh, interesting. Okay, so in fact, the glia Krebs cycle uses, as I was saying about, like an enzyme sort of binding to fluoroacetate, thinking it has another protein and being unable to, move forward in the process of changing it. Turns out that the glia actually do use a different enzyme. So the fluoroacetate does get into the neurons, but it doesn't disrupt their Krebs cycle because it doesn't bind to that specific enzyme. Okay, thank you, Christopher. And I didn't have a good answer for size during sleep or exercise, but I'm being told on the phone here that it's likely that there are no size during exercise, and that during exercising, the breaths get very large. Right now, once you start like either stressing or exercising, your entire tidal volume is going to increase. You're going to be taking larger breaths, and you're also going to start exhaling to get your breaths moving faster. So when that pattern changes, when the normal eupnea pattern changes, if I go all the way back to what the pleth, when we start to get bigger, right, it gets very difficult to identify the size. So whether or not they're there or not, I think part of that we don't really have a good answer because they're probably hard to identify. That would be my response, I think.
Sleep?
I don't know off the top of my head. No, I've seen the mice sometimes fall asleep. I've done these pleth experiments, so this is anecdotal now, but I've seen them fall asleep, and I think I still see size. Whether or not the mouse is actually sleeping, I'm not sure, but I've seen them fall asleep in the pleth, and it looks like, this rhythm changes. It gets really slow. I've sound like this, while it's sleeping, it kind of gets a slower breathing, but the size are still there. So.
With about the same rhythm?
I don't know if I want to answer. I haven't analyzed it. I haven't looked at that, so I'm not sure. But it didn't jump out at me that it was very different, not enough to go and do the analysis on it. And Christopher mentioned to me, he thinks the basal psi rate seems to be the same in sleep. So, and he has done a lot of these recordings recently. It's been like nonstop doing plethysmography. So he's been looking at a lot of this and seeing a lot of size. And so he doesn't think that it changes during sleep.
Daniel, do you know why the rhythm speeds up in vitro? Is it because there are these peptides that are being delivered in vivo that are missing in vitro?
Are you talking about the difference between the frequencies here on the left and right?
Yeah.
Are you talking about the,
Mm-hmm.
Okay. So it's actually slowing down. The rhythm slows down in vitro, right?
The size, there's one for every 8 seconds on the left, and there are 4 for every, I'm sorry, I guess.
Okay. I misheard. If that's not the, so these are actually different traces. They're not continuations necessarily. No, in vivo and in vitro, I find that the psi rhythm stays pretty consistent, despite the fact that it's moving from a living unanesthetized mouse all the way down to a slice preparation. Like I said, I've done both of these kinds of recordings, and I've seen the psi rhythm maintain about a 2 minutes to 4 minutes to kind of changes per slice. But once you kind of see the rhythm frequency, it doesn't change that much for an individual slice or animal. And so it's the same in vivo and in vitro, which is really interesting when you kind of look at the frequency here in vivo, which you see a mouse breathes a few times a second, to all the way down to the slice preparation. Now the mouse breathes, the neurons are bursting once every 5 seconds or so. And the reason that's happening is just because you're losing a lot of the mechanical feedback of the normal system that would normally be kind of like once you start breathing here, you know when your breath is full, and so your system kind of feeds back and just says like, start breathing out and then start the next step. Whereas here, there's nothing, you're losing a lot of that inhibition that's normally coming back into the system, and so the rhythm just ends up running slower. Does that kind of answer your question, Jen?
So that makes sense to me that you're missing some of that feedback. I guess I don't understand what's on this graph then. So the big peaks on the right, those are representing the size, and the smaller things are the regular breathing rhythm?
Yeah, that's right.
So the size are happening about every 4 seconds, where on the left, only the large ones are
like,
this?
No, so on the right here, this is an inspiratory burst, this is an inspiratory burst, this is an inspiratory burst, this is the psi.
I see, okay, all right, sorry.
And it's not these little things. No, that's fine. It's good to know that, these traces maybe could have been a little bit better here. Here, these are the inspiratory bursts or breaths. These are not breaths. These are the size. And I liked, I showed this figure where it kind of zooms everything out. And now you can see each of these big zoonia, and these ones are the size. And I'm wishing I had the same thing. I have it, but I just didn't put it on the plot. Same thing for the in vitro, so you can see, because it really does. In fact, when I'm doing the electrophys recordings, Sometimes I don't really know that the Psis are there because they're kind of hard to spot. You don't really know they're there until you zoom all the way out like this in vitro, and you actually will see like regular giant peaks. So either it's some random noise, or you go and look at it and you're like, oh no, those are actually Psis every few minutes. So very regular.
Are there any other questions? Whenever you're talking about this, I end up sighing. I don't know if this is happening to anyone else. It's like once you think of me sighing, I'm like,
Yeah. Yeah, I think it happens like right after I talk about the first sigh. I'm like, a sigh is a large volume breath. Everyone's like, oh. I think it's interesting for sure. Oh, go
Sorry.
ahead.
Talking about this kind of empathetic sighing, I was actually wondering if mice yawn and what the relationship between that type of deep breath, or if that's programmed, or how does that relate to sighing at all? So I guess this is a more general question about all the different types of breaths one could take.
Yeah. Yeah, I think I'm pretty sure yawning is distinct, like recordably distinct. The paper I showed at the end about the group Lee et al. In the Nature paper, I remember they gave a talk where they focused almost like half on the the actual maybe you could call it like electrophysiology, and the other half was on like the emotional connection to Psis and stuff like that. And they were talking a lot about the different connections between different breath types like yawns or gasps, and the connection with emotion. They were trying to link it. I think it was like the last 10 to 15 minutes of a presentation I saw from him. So I'm wondering if recently he hasn't published something. That would be the Ping Lee at Stanford, who has stuff like that. Yeah.
Dan, do you know if any other, when we're talking about mammals here, we sigh, mice sigh. Mm-hmm. Do other animals sigh? Other animal meaning, for example, we work with frogs.
Yeah.
Do they sigh?
I don't know for sure, but I'm pretty sure I've definitely said, I think in like papers, that all animals that breathe also sigh. But you could definitely, don't quote me on that one, but I'm pretty sure, yes, they all sigh. So I guess I have a little bit more time here. I can go into the actual physiology of what's happening. So when you're, and why we sigh. So normally when you're breathing, you're only using a small amount of your actual total lung capacity. So you're just breathing, say, like out of 20% of the whole capacity. And so as you're breathing, this sort of a little bit, the alveoli at the ends of the bronchioles of your lungs, these little units for gas exchange, you can think of them as kind of like balloons, that they can be, either completely empty or they can be completely full. And when you're only using sort of a small fraction of the total capacity, the alveoli, which are, they can, because they have so much surface area, they can condense really well to get small and then really, really big. It allows you to fit so many of them kind of next to each other. They have a surfactant around the edges of, say, like I'm imagining a balloon here, where you've kind of rubbed a surfactant around it. And that means that when you crinkle up the balloon and you're, completely exhaled, when you breathe in again, they can just kind of open up and they don't get stuck. The sides of the alveoli don't get stuck on each other, which would happen if, say, you just sort of left it all crumpled up for a long time. And that's what's happening when you're normally breathing, is that the alveoli aren't completely filled up and some of the sides are sort of stuck together. And the surfactant kind of seep out. And so what you do every few minutes is to reinflate those alveoli, redistribute the surfactant, and that allows you to, when you exhale again and you kind of, the balloons get a little crinkled up, then they don't get stuck together. And that's the function of them. I'm pretty sure they discovered them when they put people in iron lungs, where they were mechanically ventilating them, and they found that people actually needed every few minutes these large ventilations. So very interesting.
We have a huge amount of data on babies, premature infants, respiration signals for something like close to 50 babies for 5 years, something like that. And but we've never looked at the size. I know that they're there, but we've never seriously looked at them. So if anybody wants a project attempting to correlate what you're seeing with what's seen in premature infants, whether, for example, there is a development over time from the very earliest infants to later on, do the size change, or the frequency of size? Does that change, or does the amplitude get larger, or anything else? It just hasn't been studied.
Hey, John?
Yeah.
Can you hear me?
Yes.
Yep.
The shortly before birth, the compliance of developing babies' lungs are quite different than right after birth. So we'd have to probably look into how the compliance of the developing lung might be impacted by a Psi. It might not look exactly the same as a very large breath. It's definitely something to think about, though. But I know that the compliance.
We do see large breaths.
You do? Okay. I just know that the compliance changes a lot right before birth.
Yeah, not surprising.
Well, we're getting close to closing time here. It's 12:55. So why don't we thank Daniel for a fantastic presentation?
Excellent.
And
Thanks, guys.
I don't know who's speaking next week. I don't have the calendar. We'll wait to hear from Greg on that. But I hope everyone has a lovely weekend. And as always, I'll remind you, voter registration is still open, but it closes on October 13th. So if you are a Virginia resident interested in voting and not yet registered and are legally allowed to do so, this is your last weekend to register. So everyone take care, and we'll see you next week.
Okay.
See you guys.
Thank you.
Thanks.